Very good morning to one and all present. Good morning and welcome back to the ORKS Ocean Research Knowledge Series 2026. This series is a 30 plus year our scientific expertise lecture organized by the Center for Ocean Research and National Facility for Coastal and Marine Research. The Center for Ocean Research is now opening its 2-year MSc program in marine biology. Admissions are open and the last date to apply is 14th of August 2026 and the classes begin from 17th of August 2026. Interested students can reach out to the admission coordinators. We will give you all the details in the chat box. Today's lecture, we have an expert, Dr. Mrinalini Ganesan. She will be giving the expert lecture on using metagenomics to monitor ecosystem health and antibiotic resistance. We welcome you, Dr. Mrinalini Ganesan. Over to you. Dr. Mrinalini, are you there?
Yes. Good morning, everyone. I am Mrinalini Ganesan, working as a technical associate at NFCMR, Center for Ocean Research. So today's topic is on microbes as environmental guardian using metagenomics to monitor ecosystem health and antibiotic resistance. So this topic covers on 3 important topics about bioindicators, metagenomics, and antibiotic resistant genes. So why we have to monitor the monitor ecosystem health? So we already have a traditional monitoring which we are being followed for the years. Like we are calculating the recording the water parameters such as pH, dissolved oxygen, nutrients, and salinity, and also the heavy metals present in the environment like sediments and water columns or in any organism tissues. So they provide only a chemical snapshot of conditions at a single moment. So these results are based on what we collect that at the particular moment, but not a history or a history like from. It is not same as what we recorded yesterday or day before yesterday, and even if we measure after 6 hours later, then there will be a change. Biological responses to stress are often missed entirely. So we cannot capture cumulative or sublethal effects over time while using traditional monitoring. So in order to overcome that limitation, we are looking into the biological signal where microbial monitoring fills the gap. So here microbial communities shift within hours to the days of disturbance. So if we look at the last 1 week in particular environment, we will be able to tell what are the microbes present on Sunday, and we can measure the same. If we can tell if we have the same microbial community on the end of the week. So community structure reveals ecosystem stress earlier than chemistry. So they capture cumulative, like they are not capturing only at the particular moment, but a cumulative impact on the environment, not just a single reading. So what are bioindicators? So organisms whose presence, abundance, or condition reflects the quality of their environment. So whether a particular organism is present and in how much abundance they are present in what condition, like if their health condition or functional condition is normal, or some kind of function is expressed in large amount, means then that reflect the quality of their environment. So that can be a pollutant, or that can be a normal baseline, or that can be a dangerous lethal pollutants. So most of the bioindicators we are looking into are diatoms, algae. So they are sensitive to water chemistry and whatever the nutrient load present in that particular water ecosystem. Like you might have heard about eutrophication, like where the algae grow in large number when there is a organic overload. And we also look into the macroinvertebrates like aquatic insects and their larvae. So they are mostly used in the stream health indices. Like in the rivers, we are using their presence and absence to indicate their health indices. And we are also looking into the fish population, like what are the population of fishes present in particular environment, whether they are present just like right now, or they are being present in the particular environment for over the years. And based on their tissue health, like if their tissue is in normal healthy condition, then we can say that there is no other pollutants present in that environment. If there is some damage in the tissues or in their organs, then we can tell that there was some oil spill or something like any pollutant mix in the environment. So among the all these bioindicators, microbes, they respond fastest to all, like within 2 hours, 2 days. Like if there is some heavy metal overload, then the microbes are the first one to respond. When we compare to the other animals like diatoms, fish, or macroinvertebrates in the water ecosystem, so microbes respond rapidly to the environmental disturbances and are ideal for long-term ecosystem monitoring. So why microbes are ideal guardians or sentinels? So they are ubiquitously present on the earth. Like we can actually see microbial presence in soil, water, sediment, air, or any host organisms like oral microbiome, gut microbiome, organ associated microbiome, or skin associated microbiome. So they are ubiquitously present, and their growth rate is very high, like fast generation times. So high diversity and rapid reproduction allow nearly real-time tracking of the change. And they are pollutant sensitive, highly sensitive to contaminants, pH shift. Like some microbes will be thriving under some microbes will be thriving under pH 7 or 8, and if there is some pollution mix means, then there will be reduction in the pH, then that will first affect the microbial population. And community signal. So like if you are having 100 different types of bacterial population, so that 100 difference is habitat to that particular environment. If there is a shift in the community composition, then we can say that there is an indication that ecosystem health has been altered by some chemical spill or something, or any man-made chemical pollutants. So over the years, we have been culturing the microbes from sediment, water, or any organ tissues using microbiological techniques. Like we are going to do the isolation, like we are going to bring the samples from the environment, and we will doing the serial dilution, and then later we will be sticking in media specific or general media related to microbiology based methods. And then we will be doing the single colony isolation, and we will be doing identification of pure culture and staining through either they are gram positive or gram negative, and other staining like micro like bacterial specific, phylum specific class staining and all. And then we eventually do the identification using biochemical test. So like indole test, methyl red test, and nitrate reduction test, and some kind of test, catalase test, to identify what particular microbes we have isolated. Over the time, the if you look into the limitations, so only 99% of environmental bacteria are unculturable. Like only 1% of environmental bacteria has been cultured in laboratory, and they have been recorded as a database in worldwide. So about 10 to the power of 31 bacteria exist on the earth, and 10 to the power of 4 are cultured bacterial species. But if you look into the human gut microbiome specifically, about 60 percentage are unable to be cultured, or they are yet to be cultured in the lab condition. So about 60 percentage of human gut microbes biome, we have no idea what are the species what type of bacteria or what type of microbes is present in our human gut. So if you look into the human oral microbiomes, then they account for almost 50% of uncultured bacteria. So to overcome this limitation, so we are doing sequence directly from the source. Like as I said, bacteria are like any microbes are present ubiquitously in environmental sample, human associated sample, animal associated samples, and plant associated samples. So we are going to take the samples directly. Like if we are going to look into the sediment microbiome, then you are going to collect the sediment from estuary or any marine sediment, or from soil, agriculture soil or forest soil, and water if you are going to collect from, you want to look into that microbiome of deep sea. So then you are going to collect the water from the deep sea, and you are going to isolate the DNA directly from the source, and then you are going to do the sequencing. That is our advanced technique we have been doing for the last 10 years. So metagenomics is from culture to plate metagenomics, as I said previously. So less than 1% of environmental microbes can be cultured in the lab. So in order to avoid this limitation, we are doing metagenomic sequencing of all DNA in a sample directly. No culturing required. So delivers both taxonomic identity and their functional gene information. So what we do is we will collect the environmental sample, we directly isolate the DNA and from the sample, and we will be sequencing using two different methods, and then we will be doing the data analysis in bioinformatics software. So this is the two types of sequencing we mostly use for microbial database or microbial diversity studies. So one is meta barcoding and one is metagenomics. So they both are actually very different. So in the previous years, these two terms are always mixed up, and it's not clear. So here, if you look into the sediment sample, like if you are collecting an estuary, like you are collecting, for example, you are collecting a sediment sample um, from Kovalam estuary. So that particular sediment will be containing all these types of DNA. Like they contains DNA from bacteria, they contain DNA from archaea, they contain DNA from fungi, plants, animals, and free DNA, viral DNA, and any other animal, human or any type of DNA will be present in that sediment. But when we segregate this particular DNA, like we are doing the isolation, that this is the starting material. The material is the same starting material, which is sediment based DNA. In meta barcoding, we will be looking into only the particular barcodes. Like in supermarket, we will be giving barcode to each items we are going to store in our supermarket. Like if we read that barcode, we will be know, okay, this is this is belong to a scissor, or this belong to some kind of rice product. So that when we read the barcode, we will know what product that is. So that is called, here meta barcoding means there will be specific barcodes like 16S rRNA barcode, ITS for fungi. So cytochrome oxidase 1 for invertebrates, and 18S for animal, plants, and proteins. So these are some kind of barcodes we are universally using from years ago. Like from years ago, we are using these kind of barcodes to identify species. Like if it can be any species like bacteria or fungi or anything. So barcode is nothing like a target specific genes which we can use to identify particular living organism. So we design a primer specific to this bacteria, and we will be only amplifying the sediment DNA with particular 16S region. And here we will be only able to check into the bacterial diversity, not any other. If either fungi or anything is present, we cannot be identifying other fungi, other living organism, only the bacteria in metabarcoding. Um, in case of metagenomics, we will be taking the whole DNA, and then the whole DNA will be carried out for sequencing. That is called metagenomics. So after this, we will be doing the next generation sequencing process, which is which is same for both the metabarcoding and metagenomics, but the preparation before loading into the NGS, like Illumina machine, that will be different. So after that, we will be getting same kind of result, but like same kind of output. So here we can be finding who is there. In the meta barcoding, we can only identify who is there. Like taxonomic like list, if you have done 16S rRNA meta barcoding, then you will be able to identify what kind of bacteria, what species is present in the sediment we have taken up for our analysis. And in case of metagenomics, then we can be able to look into who is what is the taxonomic profile and what that particular taxonomy is functioning, like whether they are involved in any kind of cycling, carbon nitrogen cycling, or any kind of stress responding genes, or any bioremediation activities. Like that we can check in the metagenomics. And the if you look into the advantages, metagenomics provide species level classification when we compare to the meta barcoding. But one disadvantage is that the cost is very high in the metagenomics, and also the bioinformatics data analysis part is also quite tedious when compared to the meta barcoding. So we can either use meta barcoding or metagenomics based on your study. So these are the usual process we do in the metagenomics related workflow. So we have the raw data and we will be checking whether the raw data we received is good or not, and then quality filtering and identification using publicly available databases like NCBI. And then if you, after identifying the microbial population in a particular environment, we can segregate based on the environment pollution present in that particular ecosystem. Like if there is soil contamination chronically means, then that particular environment will be enriched with hydrocarbon degrading taxa. If it's related to sewage pollution means, then that particular environment will be enriched with fecal ash, like methanobacter, fecal coliforms will be present more in that particular environment. And if it is related to heavy metals, then those which can reduce the metal or oxidize the metal will be higher in particular environment polluted with heavy metals. And in case of eutrophication, those bacteria which can convert organic like organic contaminants, like that will be growing faster in particular eutrophication. Like for example, if a particular place is polluted with fertilizers, like from the runoff from aquaculture or agriculture river stream, then that particular environment will be having those bacteria which can take up the fertilizer as their feed, and then that particular taxa will be higher, but other taxa will be in less numbers. So based on this microbial signatures, we can identify what kind of pollution was chronically present or acutely present in that particular environment. So when we look into the environmental antibiotic resistance, so that is a very major concern worldwide, because before we were using some kinds of antibiotics like ciprofloxacin or amoxicillin, like something like that, penicillin. So now after this 10 years, like the last 10 years, we human has developed major antibiotic resistance towards these generic antibiotics. Now we are in search of antibiotics which are not resistant to the bacterial existing in our environment right now. So due to which there is a very crucial issue, like we couldn't treat some kinds of inflammation or any kind of infection happening in human body. So we are looking into a new type of antibiotic, because the old antibiotics are become resistant to the like the bacteria or the viral become resistant to whatever antibiotics present right now, because we are polluting the river stream from wastewater, hospital, and agriculture and aquaculture. So we are using antibiotics particularly in these sources where waters, like they are polluted with antibiotics. So that lead to bacteria adapting to these antibiotics. So they are no longer bactericidal or bacterial toxicity is not happening because they are adapted to the antibiotic itself. They can live in particular antibiotic. Even in like 10 years ago, they were able to kill the bacteria, but now they are thriving in the antibiotic presence also. So that is called antibiotic resistance, and that is what we call resistome analysis. And why we should care about ARGs? Like ARGs are present ubiquitously in every environment right now, so they can persist. Like they cannot get degraded. They can persist in longer in the environment, and they are disseminating among the environmental microbiome. Like they are spreading from one water ecosystem to the other water ecosystem, through which the antibiotic resistant genes is not confined to one particular environment. They are majorly spread throughout the world through certain sources. So transfer to pathogens. Like if this antibiotic resistant gene is transferred to either mycobacterium or some kind of Shivanella, like then they will be acquiring that particular resistant genes. Then we can no longer treat the bacteria. Like if it's TB means mycobacterium, means if the mycobacterium has gained some kind of resistant genes, then we cannot treat with the whatever antibiotics or treatment available now. Then we have to look for a new source of antibiotics that are not resistant to the bacteria. So they also create a one health implication in the world. Like the environmental reservoir, they're acting as environmental reservoirs which are directly risking the human and animal health worldwide. So if you look into the aquaculture, the fishes are affected with various bacterial infection like Aeromonas, Vibriosis, and certain other bacterial infection. Then we no longer can treat the fish that are infected with this bacteria if the antibiotic resistant genes has been acquired, transferred to this particular environment. Then there will be a huge economical loss, and even death can happen to the humans. Like there are certain records that due to the antibiotic resistance, some kind of even a small infection has led to death in humans. And so how we can use metagenomic for resistome survival is like after we are doing the sample collection and DNA isolation and then sequencing, we can use common reference databases like SCARD, ResFinder and ARG unknown databases, which has both experimentally proven antibiotic resistant genes and predictive resistant genes. These are the collection of database that has both experimentally confirmed ARGs and other predictive, like this may be an antibiotic resistant, like that they have predicted. So using this kind of database, we can find whether antibiotic resistant, like what class, like for example, if it is cephalosporin or if it is carbapenem or penem antibiotics, we can find if that, for example, if in the covalent history, if you are looking at it, we can check whether what kind of antibiotic resistant gene is present, whether that antibiotic resistant gene belong to this particular class of antibiotic or it's belonging to some other antibiotics. Then we can predict that. Then we can customize our antibiotic treatment to the patients for something like that. So that is how we use pentagenomics for resistome surveillances, like data collection, and then we are going to do the reference database based analysis. So antibiotic resistance, metagenomics will be helping in one health perspective, like animals, environment, humans form a single interconnected system. So whatever present in the sediment, that will be taken up by the lower level organisms, and then that will be entering the food chain, and eventually it will be entering the human system also. So through this, we can study the trophic transfer level, like transfer to from environment to humans. So that is how metagenomics will be useful. So this is one of our case study that was published in Frontiers in Marine Science in 2025 under Dr. M. Ravi's guidance. So we have taken up polychaetes from polluted estuaries along the southeast coast, and we have checked whether the polychaete gut microbiome carry any kind of resistome genes, and so we were able to identify that different pollution level, based on the different pollution level, the presence of ARVs also differing. And some of the key finding is like patterns linking sediment exposure to gut resistome composition. And the take home message is microbial communities are powerful, fast responding bioindicators of ecosystem health. Metagenomics reveals both microbial diversity and functional capacity without culturing. Like we no longer have to do the culturing. We can do the metagenomics isolation, metagenomic based microbial diversity and functional capacity analysis. And environmental restomes are sensitive indicators of man-made impact, and polychaete gut microbiome shows promise as a built-in guardian for sediment health. And microbiome based monitoring complements, not replaces, but they only complement. We have to combine all the three, like microbial culture and traditional environmental chemical based analysis with this microbiome based monitoring. So we have to combine three approaches while we are going to assess the environment. So this is my topic. So thank you. I am glad to answer any questions.
Thank you, Dr. Mrinalini, for giving us this elaborate talk.
Yes, thank you.
Do we have any questions from the participants in the chat box? Participants, if there are any queries, you can post them in the chat box. Meanwhile, in the chat box, few informations regarding the ongoing training programs and upcoming workshops are being shared in the chat box. For further information on analytical services, instrumentations, training programs, workshops, you can reach out to us by using our official website, or you can also follow us on social media pages like LinkedIn, Facebook, and Instagram. I see there are no queries from the participants in the chat box. Thank you, Dr. Mrinalini, for joining us today.
Thank you.
Thank you one and all for being present today. Please join us again at 2 p.m. for another informative lecture by Dr. Achyutan. Yeah, so Dr. Mrinalini, there is a query in the chat box.
Yes, yes.
But I'm not sure how this is related to this particular talk, but they're asking how, is there any diatom indicator for fisheries?
I'm actually not sure. Has been mentioned in the water ecosystem, but I'm not sure about the diatom indicator, a particular species name. I'm not sure.
Thank you, Dr. Mrinalini.
Yes, thank you.
With this, we'll close the session for today. Please join us again at 2 p.m. for another informative lecture. Thank you.